EU has MRLs for pesticides ranging from 0.01, which is the limit for baby food, up to much higher levels for some of the less toxic pesticides. The veterinary drugs have some similarities to pesticides. They're licensed and authorized chemicals. If they're correctly used, then you'll find minimal level of residues. The problems arise when withdrawal periods are not observed. So under correct use, when a sulfonamide is used, there's a period of time between administering the drug and slaughtering the animal. And that period of time is fixed so that the drug is metabolized and the residue is minimal in the finished product. Problems arise when the drug is administered and the animal is slaughtered a short period after administering the drug. So that's for permitted substances like the tetracyclines and the sulfonamides. There are some veterinary drugs that the EU does not permit under any circumstance. So malachite green is not permitted for use in treatment of farm fish, for example, and any detectable residue is an infringement of the regulation. The nitrofurans are banned substances, as are many of the hormones, which are growth promoters. In the EU, we don't permit the use of growth promoters. That is not universally the situation because, for example, in the US, natural hormones are permitted for use as growth promoters, but not in the EU. Scientifically, at the moment we tend to analyse classes of compound. Scientifically, the challenge is to bring these different classes of compound into a single analysis. There's many of the veterinary drugs that still remain very difficult. Coccidiostats are difficult. The penicillin compounds are difficult to analyse. So it's not a trivial task analysing veterinary drugs. Many of these are bound to the matrix, so you can't easily release them from the matrix. In many instances, it's not the parent compound that you look for, it's a metabolite, because the substances are metabolized. So it's a difficult area analytically, and there remain challenges here. We tend to use screening techniques in the veterinary drug area. So one of the solutions to this problem is to use a screening test like the PREMI test, which is an antimicrobial screening assay. And here you'd screen the animal products. If they were negative, you would then no longer do any further test for antimicrobial agents. They would pass. So this is a quite simple screening test that can be carried out quite rapidly. If there are positive results, you have to, of course, to do instrumental analysis. And generally in this area, if you are involved in enforcement and prosecution, you have to do LC-MSMS. It's the only way you can really convince a court that there's been an infringement of a regulation. Again, in this area, the EU has a lot of directives, so all of the residue limits are controlled for each of the different substances. So we've moved through the food chain and we're now at the factory stage. So the raw materials are coming into the factory and we're doing things to that food. We're adding value to the food before it goes off and it's packaged and goes to the supermarket. And this is the stage when there's the possibility of processing contaminants. And if I ask most people what they think about processing contaminants, they'll immediately mention acrylamide, because this was a big story 4 or 5 years ago. And acrylamide's formed through Maillard reaction, so it's formed where food components react with one another when, for example, you form French fries or you form biscuits, a number of products. So these processing contaminants are formed by chemical reactions, and they're either reactions between food components or between a component and an additive or between two additives. The nitrosamines have been known a long time. The work on the nitrosamines in the 1970s, and these are formed when you use nitrite in cured meat products and that nitrosating agent reacts with secondary amines to form a range of nitrosamines. So processing contaminants are not a new phenomena, they've been known a long time. Ethyl carbamate's been known a long time, as has HMF. Benzene can be formed by reaction with additive and food component. Furan was in the news even more recently than acrylamide, and was found to be present in bottled baby food, again a reaction product of food components. The one probably you're familiar with as well is the 3-monochloropropane-diol, the 3-MCPD, and this is present in soy sauce, and it's a reaction product where HCl reacts with the protein when you make soy sauce. And this is the only one that's controlled in the EU at 20 ppb, because it's very difficult to control these processing contaminants when they may be formed not only in the factory but also in the kitchen when you're preparing foods. We often think of food packaging materials like plastic bottles as being something that protects our food, and of course this bottle does protect the food, and it enables us to transport the food product around the world and present it to the consumer in a very attractive format. But this plastic bottle is not simply made from the plastic polymer. The polymer itself is completely inert, so the long polymer chains will not cause any problem with the food. The problem arises in the fact that you have to blow this bottle from the polymer, and in order to blow the bottle and to present the bottle in a format that it's going to serve its ultimate use, you have to add a lot of chemicals to that polymer. So when you're blowing the bottle, you need to have a release agent so that it comes out of the mold cleanly. You may want to put a UV absorber into the plastic so that if this bottle is in the sunlight, the UV light doesn't damage the colour of the food product. You may want to put an antioxidant in the plastic. So in manufacturing packaging materials in their finished form, a lot of chemicals are added to the plastic. The problem is that these chemicals are not bound to the polymer chain. These chemicals are simply sitting in the polymer chain there, and when you bring that plastic in contact with the foodstuff, if those additives are more soluble in the food than they are in the plastic, they'll be extracted into the food. So there's a large number of different low molecular weight compounds that are present in packaging materials that can migrate or leach into packaging and cause contamination. So styrene is the monomer from which you make polystyrene. So if you use a polystyrene cup for drinking coffee or tea, then there's the risk of that styrene monomer migrating from that polystyrene cup into the coffee. Diethyl hexyl adipate is a plasticizer used in cling film. And this is very soluble in fatty foods, so when fatty foods like cheese are wrapped in cling film, this plasticizer can migrate into the cheese. BADG is bisphenol A diglycyl ether. It's the monomer used to make the coatings of metal cans. You don't put the food in direct contact with the metal can. There's a polymer coating between the can and the food, and this monomer has been shown can migrate into fatty foods. And we're not just dealing with plastics. The same occurs, but to a lesser extent, with paper and board, and with all materials that are used in contact with food. Now, in the EU, we don't use the term just packaging. We call them food contact materials. It's different to the US. In the US, they're called indirect food additives. But in Europe, we call them food contact materials. And this covers not just packaging, it covers everything that's used in the home. So if you use a plastic chopping board in the home, if you use plastic knife or plate, if you use the inside a microwave oven, plastic materials, or in the factory, every stage in the food chain where materials come into contact with food are covered by EU regulations. So all of these materials are covered by a positive list, so you can only use substances that are authorised, and there's migration limits that apply to each of these substances, and we do migration tests with food simulants. And there's even directives covering specific materials. So gaskets which seal the glass lids of baby food are plasticised with epoxidised soybean oil, and there's an EU directive covering the migration of these from these gaskets. The business challenge, if I'm a food manufacturer, is how do I ensure the safety of my packaging materials? What tests can I do? And the answer is you have to use a variety of different analytical techniques. You have to use everything you have at your disposal. So far we've been talking, as we come through the food chain, with single chemical entities that we can measure, and we've been concerned really with regulatory limits. Does this food contain something above or below a regulatory limit? I want to finish off this discussion by talking about something slightly different, and I'll come back again to this afternoon. And in this area, we can't measure a single parameter. We can't measure a pesticide residue. We can't measure trace element or anything else. We're dealing with authenticity of foods. So the issue here is this food correctly labelled? And the example I want to use is the example of honey here. Now, the price of honey varies enormously across the world. There are some countries where honey is of an inferior quality and you can buy it quite cheaply. There are other places where single floral types of honey are highly prized because the flavour is really excellent and the honey is only produced in small quantities. Now, it must be enormously tempting, if you're selling honey, to blend some of the cheaper honey with the more expensive honey. And there's quite a bit of fraud in the honey area where this goes on. So the honey being sold to the consumer isn't exclusively high quality honey. It's got 20%, 30% of lower quality honey mixed in with it. Now, in chemical terms, honey is honey is honey. It's very difficult to tell the difference. So how do we tell whether this is authentic honey as presented on the label? And the answer is that we measure not just one parameter, but we measure a range of different things. So there's a possibility of measuring volatile components in the honey, isotope ratios, the sugars, free amino acids, trace elements, and so forth. And each of these parameters characterize the honey in different ways. So the trace elements in particular tell us about the geography. So the isotopic patterns tell us about the geography. So the isotopic patterns of a food product coming from Australia will be different to the isotopic pattern of foodstuff produced in Europe. So looking at the isotopic pattern tells us a bit about geography. Looking at the volatiles will tell us about the floral source. It'll tell us the type of flower that the bee went to make the honey. But it's really this slide that I want you to look at. And if we look at this part here, these are three types of honey, and we've started here to construct a database. So this circle here represents authentic honeys of a certain type. So I've been doing an analysis and using statistical techniques, using principal component analysis. I've taken some volatiles and an amino acid, and using these principal components, I've said everything that's within this circle is authentic honey of this type. And then when I analyse this sample here, and I find it's not part of this population, I can say there's a 95% probability that this is not authentic honey. So I don't have the same certainty as measuring against an MRL. This technology gives me a probability. And in the authenticity area, we tend to work with probabilities because we're using statistical techniques. And I'll come back to this again this afternoon. So to conclude, I just want to draw this together. And as we went through the talks, I mentioned different analytical approaches that you might use. And generally, we're faced with a choice of screening technologies. So some rapid method that enables us to look quickly at samples to tell whether we should investigate further. And then we have the choice of more sophisticated methods, quantitative methods, to probe and measure contaminants and residues. Now in the screening area, the strengths of screening techniques are that the cost is generally low. So if we're buying diagnostic tests, we don't usually have to pay much per test. They can be carried out very quickly. You can do the analysis on site. So these diagnostic tests are being used in the slaughterhouse to test the urine of animals for veterinary drug residues prior to slaughter. So you don't have to go to the laboratory. And you can do these tests with a relatively low skill. You don't need a PhD to do these tests. And these are all big advantages. On the downside, you need to know with these tests what the false negative rate is. So if you're screening, whether you're screening with a diagnostic test or screening with an instrumental method, you need to know what the failure rate is. And if it's got a high failure rate, then really it's not serving its purpose adequately. If you get a positive, you need confirmation, which means that you don't want these screening tests to be too sensitive. Because if they're too sensitive, you're getting too many positives that need confirmation. On the opportunity side, I think increasingly we're seeing tests that are based not on antibodies and single targets, they're based on much more broad biological activity. Calyx assay is based on receptor assay, so we can screen for classes of compound rather than single compound. Coming to the more sophisticated instrumental techniques, the big advantage is that you get unequivocal results. So if the analysis has been properly conducted with a validated method, you can feel 100% confident in the result, and you can go to court and you can defend this result. You're getting a quantitative measurement, so you're not just saying yes or no. And the other advantage nowadays is that even though this instrumentation is expensive, it's very flexible. So if I have LC-MS, MS in my laboratory, I can be doing veterinary drug testing analysis in the morning, I can run pesticides in the afternoon, and I can run mycotoxins overnight. So you can switch very quickly from one analyte to another. The techniques are very, very versatile. The downside, of course, is the costs are quite high. Running costs are not insignificant. You need skilled operators, and you need access to good service engineers to make sure that your instruments are running 24/7. I think for the future, experience has shown us that even though some of these techniques are quite expensive at the moment, the cost will come down. As these techniques become more and more used, inevitably, through economy of scale, the cost to the end user becomes less. So this is a big advantage. So just to really summarize, I started by talking about regulatory limits, and this is the basis on which we trade food, and it's the basis on which we protect consumers by saying this is safe or this is unsafe. We have an array of technologies available at our disposal, and mostly now sensitivity of these analytical techniques is more than sufficient to enable us to check foods against these standards. So 10 years ago I would have said we need more sensitive methods. Now I'm saying that I think we have more than adequate sensitivity to measure against these regulatory limits. The focus more and more is on reducing cost and increasing speed of analysis. We need to do more testing, but we don't need to spend more money. We need to do it at a lower cost and faster. We're seeing multi-methods being developed which enable